Okay, hi everyone. We are live. This is another Thursday biotech workshop. I am Danielle Snowflack. I'm the Senior Director of Education at Edvotek. And we are happy to be able to do this fun post-Halloween food science lesson with you. This will be a little bit of a training workshop that you can do in the comfort of your own home. So thanks again for joining us. It's always great to see you all here. So just again, for those of you who are not familiar with Edvotek, we are the biotechnology education company, and biotechnology includes food science, which is what we're going to be talking about today. We were founded over 30 years ago now by Dr. Jack Drichian, who was a professor of biochemistry at Georgetown University. And now if we think back to the early 1980s, there were a lot of really fantastic biotechnology innovations that were, coming out at this time period. And what I would say is that there wasn't a lot of that being translated to the biology classroom. And so what Edvotek did was it would take some of the cutting edge biotechnologies from that time, including restriction enzymes, polymerase chain reaction, agarose gel electrophoresis. It was taking these really fantastic techniques and creating versions that could be easily done in the classroom. And so that was the genesis of Edvotek. We work to translate cutting edge biotechnology into experiments and equipment that can be used in the teaching classroom. And we work with educators all over the world to help them demystify science for their students through active hands-on learning activities. And so today we're going to focus on what I think is a fun experiment. So we are going to look into food science, and we are going to explore different techniques in food, different concepts in food science with candy. So mostly we're going to be talking about food colorings. So what are food colorings and why do we use them? How are different colors created with food coloring? How can we determine whether our foods have food coloring in them? And so we're going to be using two techniques to analyze that in today's workshop. First, we're going to use paper chromatography, which uses a paper matrix and a solvent, and that's going to separate molecules based on their physical and chemical properties. And we're also going to use agarose gel electrophoresis to separate these same dyes, the same samples. And so this electrophoresis, we've covered it in a lot of workshops, but it is really a critical biotechnology technique used in labs everywhere, every day. And it's going to use electricity in a porous gel matrix to separate the food dyes into discrete zones or bands based on the physical properties of the molecule. And so this is going to include the molecule's shape, size, and charge. And so while both of these techniques are very powerful, they are actually easy enough to do in your own basement. Well, I'm doing them in my basement. They are easy enough to be done in your home or in your classroom laboratory. And so I think you'll enjoy them. So this demonstration is going to be recorded, and our slides will be available on the website. If you'd like to be notified when the slides are posted, please fill out the form that Maria has linked to in the chat box. We are also going to be offering professional development certificates to any of you who watch live. So if you would like that professional development certificate, please check that box in the form and we will send it to you. And this link will only be live for 1 hour after the presentation, so be sure to complete it before 5 p.m. Eastern Standard Time. So it's going to be about an hour after the workshop ends. So we're talking about food science, we're talking about food color. This is truly going to be a STEM lesson. So let's talk about color in itself. So for those of us who can see color, for those of us who can see, and those of us who they can then see color, we're not going to talk about color blindness in today's lesson, but it really is interesting for the genetics classroom. And so this could always be a way that you link what you're doing in biology with some of this food science. So for those of us who can see, color plays an important role in our lives. As you can see on the slide, color is everywhere, not just in our food and our candy, but companies are introducing color additives to a lot of products. So on this slide, you can see there are soaps and personal products that have been colored. Many of us will color our hair, either, browns, blondes, and reds, which are more natural colors, or on the slide, you can see there's a whole rainbow of colors that you can dye your hair. Our clothing is dyed. Artists have long used materials that are very colorful, filled with different pigments to create works of art. And even things that we, use and throw away, like paper plates and plastic spoons and forks. All of these have been brightly colored using different pigments and dyes. And so while we think of pigments and dyes as being the same thing, they're actually a little bit different. So pigments are usually insoluble, meaning we can't really dissolve them in water or another solvent. Instead, they are particulates that are going to be suspended in a liquid binder that, when hardened, allows the pigment to coat a material. So this is kind of like a car paint, or if you paint with acrylic or watercolor paint, those are actually solid pigment particles ground incredibly finely and resuspended in a liquid binder that's going to solidify when it dries. In contrast, dyes are soluble molecules that will chemically bind to a material to color it. And that's like clothing dye or hair dye. And so we're going to focus on food in this live stream, especially since we are post-Halloween, and we likely all have candy around our house. So the color of food has always been an integral aspect of the human enjoyment of food. And so even the early Romans had believed that people should not only savor the flavor of food and the texture of the food, but also to eat with their eyes. So you want it to be pleasing to look at. And so that means the food should look as appealing as it is going to taste. And so for centuries, humans have used dyes from natural ingredients to add color to food, drink, clothing, and other products. For example, saffron, paprika, and other spices were often used to give your food a yellow or a red or a pink appearance. And, today you'll sometimes see spirulina in food, and that's going to make it green, or a tomato extract or a carrot extract. And so commercially, manufacturers are also going to add food dyes, colors to food to offset the color loss due to product exposure to environmental conditions, excuse me, like light, air, and moisture. And so sometimes what you'll see is that, a vegetable or a fruit that might be in a can may have some food coloring added to it, because even though the nutritional value is still, the same as when it was packaged, some of those unstable pigment molecules can break down over time. And so some food coloring may be added to it to give it a bright color. Additionally, companies are going to add dyes to food products like beverages, jellies, pudding, and condiments to make them look more attractive to consumers. And so color is determined by the wavelengths that a material absorbs and those it reflects. And so pigments and dyes work by absorbing specific wavelengths of light and reflecting or scattering the rest of them. So an object has the color of the wavelength of light it reflects. So a material that reflects all wavelengths of visible light is going to appear white, and a material that's going to absorb all wavelengths of light is going to appear as black. So let's think about a leaf. It's something very common, and leaves are green in general. And so in those leaves, the pigments within the leaf are absorbing light, some of which is used in the chloroplast to make energy, some which is just absorbed by the leaf in general. But there's green light reflected back out. And so what basically happens is the leaf is absorbing the wavelengths of light in the blue and red spectrum, and reflecting light in the green. And that's how we see the color. And it's interplay of biology between the rods and the cones in our eyes, and the transmission of that signal from the eye to the brain to allow us to perceive color, and the physics, which are the property of light and how they create color. This interplay is fascinating. Artists and scientists both have studied the interplay of color and how we perceive it. And so it's interesting because colors interact with each other in different ways, basically when they're mixed. And color theory is this art and science of color mixing and the visual systems, visual effects of specific combinations of colors. So colors can be either additive or subtractive, and you may have heard this because this is the difference between CMYK and RGB, which are two common color systems that are talked about in color theory. So additive colors like the RGB system work by adding colored light to black. And that's like what we see with our computer monitor. So when it's turned off, there's no color. But when we start adding color through the pixels, so either red or green or blue, those wavelengths of light add, and then our eye perceives a different color. In contrast, subtractive colors work by removing or absorbing colors from white, and that's what we see with paint. As we add different colors to white paint, they are going to absorb the light, and we can only see what is reflected back at us. So colors are going to interact with each other in different ways when they are mixed. And so you can actually use a color wheel like this one, which I happen to have at home, to look at those interactions, but you can also see the color wheel that I have on the slide. And so colors interact with each other in different ways when they're mixed. And some of this is based on whether a color is warm or cool. Warm colors are going to be these colors on this side of the color wheel that are like red or yellow or orange in color. And so those are generally considered to be warm colors. In contrast, on the other side of the color wheel, we have the blues, the greens, and we have the blues and the greens and the purples, indigo, violet. Those are all going to be our cool colors. And we can have warm blues and cool reds based on the undertones and the way the colors are mixed, because color is a spectrum and all the colors are going to be relative to one another and relative to how they're mixed. And so, again, we can have cool reds and warm blues based on where they are on this color wheel and what colors we mix to them. So if we take a warm red orange and mix it with a red, we're going to get this cooler purple. If we take that red violet and mix it with a blue, we're still getting purple. So there's a lot of different ways to mix the colors. And so if we mix colors from opposite sides of our color wheel, so if we take, our blue and we mix it with an orange or a red and a green, they're actually going to kind of cancel each other out in terms of the color. They're either going to add or subtract out the wavelengths of light, and we're going to get a neutral shade, either kind of a gray or a brown. But it's interesting because when they're placed next to each other, they look great together. They kind of pop. They have a bold interaction with the colors. And they pop because they're stimulating different parts of the eye. So different rods and different cones. And we're going to see a strong contrast. And these are called complementary colors because they create the strongest contrast between colors. And so you'll often see sports teams use complementary colors in their uniforms. And two examples are like the LA Lakers and the New York Knicks, where we see blue and orange for the Knicks and yellow and purple for the Lakers. And those colors really stand out. So colors are also influenced by their surroundings, and this is something that I always thought was cool. So you see we have these 4 different groups of shapes next to each other, each rectangle with a colored rectangle within. So believe it or not, the color in the center of each of these pairs is actually the same. So if we look at that blue and that orange, and those are blue and orange complementary colors, when we put that shade of brown in the middle, it looks different depending on what's on the background. And that's the way our eyes perceive the way the colors interact with each other. And so this is often used by artists to create drama or illusions in their artwork. And then furthermore, because of the qualities of pigments, paint color mixing does not always match digital art. So with digital art, you can very precisely choose the colors that you're using in your artwork. But traditional pigments are not perfect. Many of them are minerals mined from the ground, which leads to interesting mixtures and colors that should not be possible due to imperfections or contamination that changes their color. And so the use of color in the laboratory is actually a great way to integrate STEAM into your classroom. All right. And so what is STEAM? So now STEM has been a buzzword for such a long time in science education. STEM lessons are those that integrate science, technology, education, and mathematics into an interdisciplinary unit. In my mind, though, I call this good science, right? Because biotechnology does not exist in a vacuum. This is an example of the process of good experimentation where science, technology, engineering, and mathematics all depend on one another. And so STEAM is actually going to take that one step further. By integrating the arts, which is the A in STEAM, we can better break down complex questions, challenges, problems, or even just global issues into its component parts to discover the nature of the problem and its relationships. This involves reading and researching not just the hard scientific literature, but sociology and the human implications between a given topic. For a lesson like we're doing today, we can talk about the interactions of colors. We can look at the great masters of painting. to see what they did with color. And then we can identify and understand the content knowledge and use a line of questioning and reasoning to generate necessary solutions to real world problems and answers to complex questions. And by integrating the arts, we change the way we look at the data, the way we approach problems, and the way we communicate the results. And so biotech is great to teach STEAM because the experiments really integrate all of these areas, especially these experiments. And the most important thing, though, when talking about these interdisciplinary experiments is that students are going to make real-world connections between science concepts in the classroom, its practice in the real world, and the importance of arts as a means for communication. All right, so let's get to our candy. So to analyze the colors in our candies, we need to remove the food coloring from our candy. And so what I am going to do is let me switch over to my cell phone. I am using my cell phone as a camera, but let me change over to my camera display so that you can start to see what I'm doing and how I remove the colors from the candy. And so to analyze the colors in our candies, we need to remove the outside food coloring from our candy. And so this is easiest in candies that are coated, where the color is a layer on the outside of the candy. And so I have some, I prepared some ahead of time, so I have a bunch of colored solutions here. But I'm going to prepare two different reds for us right now while we're looking. And so we need to remove the outer layer of the candy. And so this is easiest done in candies that are coated, where the color is a layer on the outside of the candy. And so today I'm going to be using Gobstoppers and M&Ms because those are candies that both have a colored outside layer and they happen to be the one that I had at home. And so I was curious, we have these two different red candies. Do they use the same red dye? How is it going to look on the gel? Am I going to be able to extract the color the same way? And so what we do is we're going to add this DNA extraction buffer. We're going to add this dye extraction buffer to both of our samples. And so this is going to be included with the kit. So this is the dye extraction buffer. It's included with the Edvotek kit S47. And so that is the kit that we are covering today. It's going to provide all of the reagents that you need for electrophoresis, for the dye extraction from the candy, the pipettes. It's not going to include the paper. The paper chromatography is a little additional activity that I decided to throw in for today so we can compare the techniques, but that's easy enough to do with paper that you might have at home already. And so what I did is I added my dye extraction buffer to my candy. And you kind of need to swirl the samples. You want to swirl the samples so that the surface of the candy is in contact with the buffer, and we're slowly dissolving the food dye from the candy shell into our dye extraction buffer. And so our dye extraction buffer, it's going to basically help us dissolve that color, and then it's also going to be useful for electrophoresis. Because it's going to make our samples dense. And we need that density so that our samples do not come out of our wells when we are doing electrophoresis. And so let me keep spinning this. I think I got enough color off of it for now. It looks good in red. The longer you do swirl, the more color you will get off. So you can see the color of my candy is getting lighter and lighter. You can definitely see the difference here with the M&M. You can see we've gone from a bright red candy shell to kind of a salmony pink color. And what I'm going to do is I have separate tubes that I put aside for both of these. Just so I can keep my food dye straight, I am going to remove the dye from this tube, from this cup, and place it in the tube. Now, you don't need to do that. I'm just doing it so that I can keep my sample straight because I'm going to be loading them at two different times. But you might, use one cup, rinse it out, put it in a tube, or you might just decide to use one of the cups. We include several with the kit. All right, I am going to finish getting some of this red dye off of this candy. And you can see, again, we have this bright color in the sample. I'm going to just use some clean water to rinse out my transfer pipette. For this, we're not really going to worry as much about cross-contamination. It is an area where you could contaminate, but in this case, the contamination will not cause a problem with the experiment. So what I'm going to do, I'm going to put this in the tube, and then I have my red gobstopper dye, my red M&M dye, and then I actually have some other colors of candy that I did as well. We have a blue M&M, a purple gobstopper, a brown M&M, which I'm really curious about, and then we have green and orange. So we have more samples than we have wells, but you know, I'm gonna, if anybody has one that they really want to see, let me know in the chat window. But you know, this is basically the process through which we extract the dyes for analysis by both paper chromatography and DNA electrophoresis. You can see here, I don't have a test tube rack at home, so I just use this Play-Doh as a test tube rack. All right, so let's go to our next slide. Let me make sure that advanced. Okay, it did, good. Okay, so this is the fun part. So which dyes are gonna create our colors? I'll talk more about the FDA approved food dyes in the upcoming slides, but we can talk about and we're going to talk about what colors we need to mix to get these different colors. But so for example, green, we know that's a mixture of blue and yellow, and orange is yellow and red and blue makes purple. Brown is going to be a mixture of multiple colors to form this neutral color. And we have two reds here too. So are they the same red? Are they different reds? And we're going to analyze what is in these samples using these two different techniques, which is paper chromatography and electrophoresis. Both separate the samples into their component parts, but with differences. So paper chromatography is going to use a paper matrix and solvents to separate the molecules. Agarose gel electrophoresis is going to use electricity and a porous gel matrix to separate the food coloring. But both techniques are going to separate the dyes into discrete zones or bands based on the physical properties of the molecule. And so we are going to do our paper chromatography experiment. We're going to set up for it first. So I have a piece of filter paper here. And what I'm going to do, into a rectangle, I'm going to use a pencil to mark a baseline. And you probably want to use a ruler or two to make a straight line, you know, especially if you're going to be doing the mathematics later, because you want an even baseline across the bottom of the paper. And then we're going to separate the ink. You definitely want to use a pencil when you're doing this and not a marker, because most markers will actually separate using chromatography. And so we don't want to interfere with our color chromatography. And then we are going to take one of these mini transfer pipettes and we are going to spot some of the different dyes onto this paper. And so what I'm going to do is I have some water. I'm going to clean out my pipette between spots. And then I am going to just use my transfer pipette. And with these transfer pipettes, if you need to make very like small, fine adjustment measurements and just spotting, instead of doing the top here, I actually find if you squeeze the barrel underneath, it's going to allow you to make smaller spots and have a little bit more control. And so let's spot our red dye here. And this is our M&M. So I'll put a spot there. And then with my pencil, I'm just going to put an M. And now I'm going to use the water to rinse out my transfer pipette. I'm going to try the red gobstopper next because I'm so curious to know whether they are the same or different red dyes. Because in the tube, right, they do look a little bit different. And spotted here, they kind of look a little different as well. So let me put this G here. And then let's do a purple. Rinse out between samples. Again, our purple should be a combination of red and blue. But is it going to be the same red as these other reds? You know, is it a different red? What's the proportion of red to blue? So I'm going to just do P here. And let's see, let's do blue. And if anyone has any questions, please put them in the chat box. You know, I would love to hear what you're thinking about this experiment. Do you plan to use it? Are you a parent looking for something to do at home with your kids? Because this is a great experiment for that. You know, I'd love to know what you're thinking. And then let me put the green on here as well. So we know green is a combination of yellow and blue. And so I'm just going to spot that here. And so I tested some of these colors, not all of them. So I know what'll happen with some of them, but I'm going to be just as surprised as you are. And so the blue and the green come from M&Ms, the purple comes from a gobstopper, and then we have our two different red dyes. So what we're going to do is we are going to let this dry for a little while I load my electrophoresis gel. And we're going to actually run both of these experiments at the same time. All right. So overview of electrophoresis. If you're interested in an in-depth electrophoresis discussion, definitely go to one of our previous live streams. I'm not going to dig into the specifics today simply because we have covered it before, and likely all of you are electrophoresis pros at this point. But what I've already done, I casted my agarose gels, which is like our science jello to physically separate our samples. My gel is in the gel chamber with the electrophoresis buffer. And, we're going to load it using a micropipette, and then we'll turn on the current to separate our samples. Okay. And so you should see my slides advancing. We had a problem with that last time. If you don't see my slides advancing, if you don't see my camera, let me know and I'll fix it. Okay, so what do we need to analyze these samples using electrophoresis? First, we need our samples. You may think of electrophoresis as being something that you're going to use to separate DNA, RNA, protein, but we can use it to separate dyes too. It's a very versatile technique. And in this experiment, we're analyzing our candy dyes. And I'm going to start loading them now. Where are my tips? Oh, there they are, hidden under some of my results. So don't want to show you those yet. And so here we go. I'm going to load them into the middle here because we don't know whether these dyes will be positively charged or negatively charged, and I'm loading 35 microliters. And you'll notice that as I load them, the samples are going to sink into the gel. That is because our candy extraction buffer, again, that is the buffer that comes with this experiment, the dye extraction buffer, that's going to have glycerol in it, which is going to make it heavier than water. And so the samples are more dense, they sink into the wells. If we didn't have that, and this ensures that our samples get into the wells so that we could separate them using electrophoresis. If they were just in water, likely our samples would not sink into the wells and stay there, and we would not be able to analyze them. All right, so here's my red gobstopper. That's going to be my second sample, and I'm lining them up outside, out of the way, so that I can keep track of the order. Ideally, you would be writing this down in your lab notebook, but I'm not. So, anyone have any preference in what color comes next? Let me know. I'm going to do brown next because I'm super interested to see what brown looks like. And so agarose is, oh my goodness, I just bumped that camera. So agarose is our matrix for separation. This we can think of as a scientific jello. And when I was making this gel, my son came and started trying to squish it, and he thought it was so much fun and so squishy, and it felt just like jello, which it does. It's like a scientific jello, and this is going to serve like the matrix, which is going to allow us to separate different molecules. All right. Next, let's see. I'll do purple. Why not? This is, again, the purple gobstopper sample. This, again, we know purple is blue and red, but what color red, what color blue are we using in here? And then let me change things up a little bit. Let me try orange. I'll do orange, why not? And then blue or green. Does anyone have a preference what my last sample should be, blue or green? Well, I'm loading this orange. Again, orange is going to be a combination of red and yellow. And again, this is coming from M&M, so you can see that some of these are kind of cloudy and opaque. That's going to come from like the white layer that is there in the M&M. And since no one has a preference, let me do blue. Why not? I like blue. It's my favorite color. This blue, M&M blue, is particularly pretty. So let's do that. I'm going to load that in there. So we load our samples into the gel. We have electrophoresis buffer in there. Electrophoresis buffer is going to allow the water to conduct the charge through the gel, and we need that charge because that charge is going to push the dyes through the gel. With the kit, you will get the agarose and the electrophoresis buffer as powder and concentrate, and you'll make the gels yourself. So I've already place the gel in. This chamber is going to be wired at either end with the terminals that are connected to the leads. The leads are plugged into a power supply, and that's going to generate the current, which allows us to separate our dyes. So I'm going to place the lid on here. I'm going to place the lid on. This is a safety cover. You can't stick your fingers in there when you're running the gel. And then I am going to turn on the electricity. And so this electricity is going to run through the gel and start pushing or pulling the dye molecules from one side of the gel to the other. All right, so our gel is loaded. Our electricity is on. Let me just tell you a little bit about the electrophoresis chamber I'm using. Again, if you want a more in-depth description of the equipment I'm using, we have a lot of live streams that you can refer to. You can also go on to edvotek.com to check out the equipment. I have two of our more popular electrophoresis systems on this slide. The unit I'm using is the M12, which can run up to 2 gels at once. If we were in a teaching lab, this could accommodate 12 students, which would be 2 groups with 6 students each. If you're looking for an all-in-one solution, so that's going to be your electrophoresis chamber, your visualization system, and your power supply all in one, we've just introduced the Edvotek Edge. And so we have several other options as well. You can check them all out on our website or see them in action on our live stream. So let us, why isn't my slide going to the next? I must have a link in there. When I clicked on my slide to advance it, it opened up another browser window, which kind of freaked me out. But you're all still here, which is good. Okay, so performing paper chromatography. So I'm going to put aside our electrophoresis chamber for a minute. I'm going to get our paper chromatography experiment set up. And then once that's set up, we will talk about food science. And the reasons why we have protective agencies for food and so forth and so on. So here I do have a salsa jar. I am using this as my vessel for our paper chromatography. You're going to pour solvent into the cup. I'm using a solution of 60% isopropyl alcohol here. But a great STEM inquiry-based addition to this lesson would be to have your students take their dye samples and compare the differences between solutions with different ratios of isopropyl alcohol to water. So the molecules will migrate differently depending on the proportion of alcohol to water. Additionally, you could actually change the solvent as well. So you could use salt instead of isopropyl alcohol. So this is a pretty easy setup. We have our jar with our thin layer of alcohol on the bottom, and we are going to kind of stand this paper in here so that the bottom of the paper is in the solvent. And let me tilt this a little so that you can see it a little bit better. And then what happens is the solvent is going to diffuse through the paper and moving the dye molecules through the paper. And then at the end of the workshop, we will analyze the results. So let me see if I can get both of these kind of in the screen at once. I'm going to move the gel back in. I probably can't. If anyone has a preference between gel or paper, let me know. Because I can't really get both of them in at once. And I can switch back and forth if we're curious. But what you should already be able to see is that some of our dyes are separating here on the gel. We can see here that the dyes are already starting to move through the paper. So these are cool experiments because you can really see them happening in real time. So in paper chromatography, just a little bit about the science. We have our stationary phase, which is our paper. Again, here's our paper. And you can see, you can't really see, but trust me, we have paper. And the solvent is going to be drawn up through the paper through capillary action. Our mobile phase is our alcohol. And as the solvent travels up the chromatography paper, it brings the dyes up the paper. More soluble dyes are going to travel up the paper with the solvent more quickly, and insoluble dyes are going to stay at the bottom of the paper and move more slowly. And so the solubility of these molecules is definitely based on the solvent. Again, I'm using a 60% isopropyl alcohol solution here, but if you change the percentage of alcohol or use table salt, you would get different results. And so we analyze the migration of our dyes through the paper by calculating the retention factor. So here's where we get some mathematics from STEM. And so the retention factor is the ratio of the distance the solute traveled to the distance the solvent traveled. So how far the dye traveled to how far the solvent went through the gel. So when the experiment is finished, we can calculate these values for each different dye, and it helps us analyze our results. Okay, so let's talk about food dyes, the regulation of food dyes, and a little bit of food science. And so the Food and Drug Administration was founded to protect people. And so as the use of food dyes and different commercially created food products continues to grow, concerns about the addition of food colors to food products have emerged. And so there have been discussions on whether food dyes pose several health concerns in people, and we've certainly seen this in the past. So, for example, in the late 1800s, early 1900s, a green food dye containing arsenic was a common additive to foods, and lead was actually used in candy and cheeses to color it. So people knew these things were not good for you, but there were no laws to prevent them from from use. And so the consumer had no idea what they were getting. And so in 1906, the Pure Food and Drug Act was passed. And this led to the first law that was aimed to protect consumers from commercially prepared foods. This led to the creation of the Food and Drug Administration. The main purpose of the FDA was to prevent adulterated food and drug products from being sold around the country, and it allowed the government to inspect food for safety as well. But checking stuff after the fact wasn't enough. And so, unfortunately, we found that out the hard way, because in 1937, a medication was created and the medication itself may have been safe, but the problem was that the medication was dissolved in a chemical called diethylene glycol. It needed to be dissolved in that specific molecule. It was not soluble in others. And it wasn't really common knowledge at the time that this chemical was toxic to humans, but there were no rules in place to protect people. So they didn't need to test the formulation on animals or people or in any cell culture systems before putting the drug onto the market. And so it wasn't known that this chemical, this diethylene glycol, was toxic to humans. And so soon after the drug was introduced to people, more than 100 patients died. And this was because the medication was not tested, again, before it was put on the market. So in response, the FDA was notified and extensive search was conducted to recover the distributed medicine. And this led to the United States Federal Food, Drug, and Cosmetic Act, which is the FD&C, which you have seen probably in a lot of different places. And this is a set of laws passed by Congress in 1938, which gives authority to the US FDA to oversee the safety of food, drugs, medical devices, cosmetics, and more. So let's talk about the molecules we're separating here. Let's talk about the actual dyes. And so you can actually see we're getting some good separation already here. I can see on my paper we're getting separation as well. And I will show you that in a little while because I don't want to disturb it. But you can start to see the colors separating here on our gels, which is pretty cool if you ask me. And so what we see on this slide is the structure of two commonly used food dyes. So yellow 5 or tartrazine and blue 1, which is also called brilliant blue. And at first glance, they're kind of similar molecules. They've all got rings, side groups, they're organic molecules with charge. But you can see that they have different bonds. The chemistry of these molecules is actually different. There are different bonds, different functional groups, and all of which affect the charge of the molecules. So both of these molecules here do have negative overall charges, but by virtue of the side groups and the bonds, tartrazine has a longer negative charge than brilliant blue. So we would expect that those two dyes would migrate differently in our experiment. The larger negative is going to move towards the positive pole more strongly. And so today, the FDA is responsible for regulating color additives like the ones that we saw on the last slide, which are used in a variety of food products in the United States. And so on the slide, we can see the main FD&C colorants used today. Colors for use in foods are classified as the color additives allowed for use in foods today are classified as certifiable or exempt from certification. These color additives are man-made and they've been tested by manufacturers and the FDA to ensure the quality and the safety of the color additives. And so this table illustrates 7 commonly used food dyes in the United States. Of these, blue number 1 and red number 40 are the most common blue and red dyes, while green 3 and blue 2 are rarely ever used. And so food containing these approved dyes are called for coloring food and have the abbreviation FCF preceding their names. And so on the slide, I have pictures that I just really loved because they showed foods that were colored with these different food dyes. So we can see, blue Gatorade, blue sports drink, and we can see orange nacho chips, red stain pistachios, which I will stain your fingers and your face, and it's part of the experiment of eating it. In fact, I remember reading about a study where people didn't think Cheetos tasted as good when they didn't use the food dye that stained their fingers. So again, this is going to talk about the way we see food and the color that we see food is associated with our experience. And so I just love these pictures because they did a great job showing what foods were colored with different dyes. And there are actually two more food dyes that are approved for human consumption, but in very limited applications, and so I didn't put them here. And so in foods, there's going to be color additives that are also exempt from certification, and those are going to be derived from natural sources such as vegetables, fruits, and minerals. And you'll find that some foods and clothing are colored with natural colorants. So here are few common colorants that are used in different contexts. You'll definitely have heard of indigo, which is the plant-based dye that is commonly used to dye denim. And one common controversial coloring, which we see on this slide, is cochineal. And so this pigment was in the news a few years ago because of its use in a Starbucks drink. And you would think it would be okay because it's natural, but this pigment is actually extracted from bugs. And so a lot of people are grossed out by that. The pigment itself, it's natural, it is completely safe, and it's been used in food and makeup for centuries. It's 100% natural. And so don't forget that some parts of the world, insects are a regular part of the diet. Crickets are actually a great source of protein for many people. However, in the United States, it's not common to eat insects. If you are vegan and you don't eat anything that has been living, like any living animals, you may be opposed to it ethically because of your diet. And because it's not common in the United States to eat insects, this caused quite a bit of controversy. But at the same time, it is safe and it is natural. So, every person needs to kind of weigh how they feel about this natural dye, on their own. And so for our experiment today, the mixing of colors of these dyes is an important key concept for your students to understand. This is getting back into the arts again. So we know that red plus yellow equals orange, green and yellow, yellow and blue is green, and so forth and so on. And so with food coloring, we're working with that limited palette of colors based on the approved food dyes. And so here is a color mixing chart from a food coloring company. And you can see here that each color has its own recipe, and each color uses the same colors in different combinations. And that's going to give us different final products. And so not only do the food dyes themselves matter, but the proportion of each food dye matters. So we can see green versus teal. So to make green, we're going to use more yellow than blue. To make teal, we're going to use more blue than yellow. And so we're starting with the same two starting materials, but the proportion in which we mix them is going to make the color look different. And so in addition to our chemical analysis, and so we know this is an artistic analysis based on color mixing, where we can kind of reason things out by knowing what their component parts are. But in addition to our chemical and our artistic analysis, we can use a little detective work too to figure out what food dyes are in a food product. And that is by looking at food labels. And so here we see food labels for M&Ms and for goldfish. And so companies are required to list food dyes in their list of ingredients. And so that we can see from these labels, the goldfish in particular, they use natural food colorings. You can see beet, watermelon, paprika, turmeric extracts present that give the goldfish their colors. In the M&Ms, we are using a combination of different food, artificial food colors, including blue 1, yellow 6, etc. And different combinations of those colors are going to be what gives you the final colors on your M&Ms. And so between our knowledge of color mixing and reading the labels, we can start to make educated guesses as to what colors are going to be used in each candy. So We are almost finished with our electrophoresis experiment. We've been separating our dyes for a while now. And what I kind of want to do is just go over, do a recap, talk a little bit about what's going on in the gel, and then we'll analyze our results. And so, as you remember, before the workshop, I cast the agarose gel. That's our scientific jello that's going to help us separate the molecules. We placed the gel into the gel chamber where it was covered with electrophoresis buffer. We loaded our samples. We created our dye samples, loaded them, and placed them in the electrophoresis chamber, and turned on the power supply. And that generates the current that's going to push our dye molecules through our gel. While we were discussing our dyes and food science and the FDA, the dye samples were separating into the different bands based on their charge. And so let's talk about what's happening in the gel. So electrophoresis is going to separate our dyes by charge. And so what does this mean when we add the samples to the wells and add current? So our dye molecules have overall net charges, which are going to influence their movement through the gel. Because molecules with different charges travel at different speeds, they become separated, and that forms discrete bands within the gel. Some of the dyes are negatively charged, like DNA, and they will move through the gel towards the positive electrode. Dyes with greater net charges will be more strongly drawn towards the electrodes. So if we looked at the two charged dyes before, that was our blue, brilliant blue and our tartrazine, they're going to migrate differently. And because of the larger charge, the tartrazine is going to move through the gel more quickly. So that one will be further down on the gel, closer to our terminal than the other dye. If we have any positive dyes, and looking quickly at our samples, I don't see any positively charged dyes here. But if we did have a dye that was positively charged, that was actually going to travel through the gel in the other way towards the negative terminal. So positive gel will go towards the negative dye goes towards the positive terminals, and that's separating them into different bands based on their charge. And so if you're used to seeing DNA gels, it would look like the samples are running in reverse, but the dye would be doing exactly what it should be doing based on its chemical properties. And again, the candies I used today, I used artificial food coloring. If you used candies that are colored with natural food coloring, you might see something different. And so, this is a place where it's ripe for experimentation, either at home or in the classroom. So let's start to analyze our results. I'm going to stop our gel. I'm going to move it over to the side while I'm letting our gel, I'm going to get the gel out of the way. I'm going to bring up our True Blue 2 transilluminator. Now, you might think of this as being used for cyber safe, which it definitely is, and it is a fantastic transilluminator for exciting fluorescent dyes and getting them to glow bright green. The nice thing about this transilluminator is it actually also has a white light feature. So for experiments like today, we can use the white light feature to help us visualize our electrophoresis results, our dye electrophoresis results. So I'm just going to take the lid off of the gel box. I'm going to take our gel and place it on the transilluminator. Oop, wrong way. And what you can see here is our gel. You can see our colors separated. And let's go to our analysis slide. And so is the analysis of our candy, let's do some results in conclusion and debrief on what we see here. And so was the hypothesis about our colors of our candy correct? Well, we didn't really make any particular hypothesis. We did talk about purple here as being a combination of blue and red. And what you can see is there's a different proportion of blue and red here, which did give us a different color. We can see in our red dyes, my guess is that probably the same red dye, but used in different amounts. So you can see here, this is our M&M sample. There was a ton of red. Let me just double check that that was our M&M sample. Yes, this was our M&M sample. You can see there's a ton of dye there, very dark red. Here you can see in our gobstopper, it's not as dark. There's a little bit of streaking. There could be a second red dye. So my guess is that there's a second red dye that runs right around there. We can kind of see it in our purple, and this band is kind of wide, so we might have 2 red dyes present there. Here is our brown, and if we look at our brown very carefully, we can actually see a little bit of 4 different colors. And so I don't know if this is going to come across on the camera because it's pretty subtle. I'm going to use this pipette tip to point, but we can see there's a yellow here, a light yellow. We can see this same red here. Then we can actually see kind of like this orangey yellow color. So that was our orange M&M. Yes, that was our orange M&M. I'm just double checking again. Kind of runs around here, around the same place as the red. And so we can see there's this kind of yellow there and blue. So our brown M&M is actually a combination of light yellow, red, this orange yellow, and blue to make that brown color. So again, that goes back to color mixing, how we mix all the colors together to kind of create this neutral shade, our complementary colors. And that's how we get a brown without being able to use brown. And then here we have our blue, which is just a single blue, our orange, which is a single orange yellow, the purple is the combination. So that's pretty cool. I'm super excited by the way that brown turned out. I didn't know what to expect, and the experimentation at its best. And again, you know, you don't need to have this light box to be able to visualize the results, but I bet without it, I wouldn't be able to have seen that subtle yellow color, which really just added a lot to the analysis experience here. And so again, you know, each dye traveled through the gel in a different way, and that's based on its charge and its chemical properties. So let's turn this off and move it out of the way so that we can look at the results from our paper chromatography experiment as well. Oop, my true blue is getting caught. So let me just put that here on my lap for now. You know, working in tight positions. Here we've got the paper chromatography. Again, this is just using a piece of filter paper. I did use a DNA extraction buffer to get the different colors off of the candy, but you can probably do that just using water. And you can see my dyes separated here. Again, here we see the difference between the M&M and the gobstopper. We can see again the gobstopper is lighter. And we even get a little bit of something insoluble hanging out there. So I wonder if the gobstopper does have a second red pigment, which may be contributing. A purple, again, we can see a darker blue and a red. Our blue is just that bright blue, and our green is the yellow and the blue. And so this would be an area with this experiment in particular where you can have you're going to have so much dye left over. You know, you're only limited by the amount of time you have and the number of jars. So again, I used a 60% isopropyl alcohol solution. But here's an example that I did last night. The difference between a 60 and an 80% isopropyl alcohol solution. And you can see that just by changing the percentage of alcohol that we used, you know, we were able to separate the molecules differently. And so you could use that to further look at what you're doing. Again, we mark this top. This is our solvent front. You can see I've marked them here. And to calculate like the RF, what you would do was kind of circle the middle of the spot. So here we have our yellow spot, our blue spot. Here we have our orange spot. You would take your ruler, measure to the middle of these spots, and then you know the distance that the solvent front went to. And you would calculate the RF based on how it migrates. So does anyone have any questions? You know, now is the time to ask them. I'm going to wrap up this lesson, but you know, right now would be the time to ask any questions so that I do answer them before we end. Okay, so just in my recap, again, you know what? I've got these here. I'm not going to put the true blue back out, but let me just put the gel back up here. It's not going to look as bright as it does with the true blue, but that's okay. You can still see it. It still looks pretty cool. And I dropped some candy samples on the bottom, but that's okay too. If you haven't yet, please fill out the form if you'd like to receive the slides after the presentation. I'll send you the slides and the link to the video. So just to recap, today we have covered a lot of information over the course of this experiment, but I truly think that the use of color in food is truly a great way to integrate STEAM into your classroom, which is basically STEM plus art. The most important part, though, when talking about these interdisciplinary experiments are the connections that your students can make between science concepts in the classroom, its practice in the real world. So if you think about the case of the medication that was killing people, that is science, that is policy, that is government. So the talk of the implementation of laws based on what scientific evidence shows. And so these are really great ways for your students to make combinations between scientific concepts in the classroom, its practice in the real world, and the importance of arts as a means for communication. We talked about the properties of light that create different colors and how through mixing colors, we can create new colors and influence surrounding colors. We learned about how agarose gel electrophoresis and paper chromatography can separate molecules into discrete bands based on their physical properties. And so while these experiments are fairly simple, we just did them together in my basement. You can easily do them in your classroom with your students. And these techniques are very simple, but very powerful when it comes to rapidly separating molecules from one another, again, based on their physical properties. And again, this lesson, we can not only apply science, art, mathematics, technology, it's all here and with stuff that you can probably, you can do this experiment with stuff you probably have at home. No questions? If anyone has a question, let me know. But we will be posting the presentation and the slides in the next few days to our website at edvotek.com. If you're watching this after the fact and you have any questions, get in touch with us. You can call us 1-800-EDVOTEK. You can contact us, info@edvotek.com. We're always available by email. You can contact us through one of our social media channels, through Facebook, Twitter, or Instagram. You can find us here at YouTube, youtube.com/Edvotek Inc. We do run these live streams. We try to run them every other week. So if there are any other topics you would like to see us cover, mention it on that Google form, send us a tweet on Twitter, and we'll try to incorporate that into our next lesson in 2 weeks. And we're just really grateful that you joined us for this live stream. If you need us, please reach out at any time through Edvotek, info@edvotek.com or one of our social media channels. We look forward to working with you to help you get biotechnology into your classroom. And so thank you again for joining us. Stay safe, stay sane. Thank you for joining us, and we hope to see you at a future live stream. Have a great afternoon. Bye now.